My name is Spencer, and I'll be presenting SCOTCH, which is a tri-factorization approach for identifying cell types and driving genomic features. And so, as many of you know, with the explosion of single-cell genomics, there's been an influx of data that has allowed us to understand biology at an unprecedented level. And so one of the main challenges that we face is identifying different cell types and cell states from this data. And this classification task can be useful to understand multiple different biological processes, including fate decision, differentiation, and disease development. So briefly, the task of cell type identification is broken down into two steps. First, cells are grouped into clusters where they share similar expression or genomic feature profiles. And then these clusters are annotated using some sort of marker feature or database. And there are both supervised and unsupervised methods for doing this approach or for identifying different cell types. But today I'll be focusing on an unsupervised learning approach, which is important when we consider cell types that come from samples that haven't been observed previously, such as novel retinal organoids or other more rare cell types. And so a typical workflow for the cell type identification pipeline begins with a cell by genomic feature matrix, which is then embedded using some sort of dimensionality reduction strategy, and then clustered. And then these clusters are annotated using some marker genes, usually from a marker gene list. And these genes, and first we need to identify which genes are important for each cell type. And these genes are typically identified using something like differential expression analysis. While differential expression analysis is a powerful tool for identifying these marker features, it generally misses combinatorial relationships that can exist when cells from different clusters have shared genomic profiles. And so to overcome this, we aim to identify these genes directly during our cell factorization or dimensionality reduction, such that these genes drive the different clusters. And so we've already seen one way to do this yesterday during one of our keynote speaker presentations, which is non-negative matrix factorization. And so with non-negative matrix factorization, we have an original data matrix X, which is factorized into two lower dimensional matrices U and V, such that the product of those two factors recapitulates the original matrix. And generally, these lower dimensional feature embeddings can be used for downstream tasks such as clustering. Here, in our formulation, we include a regularization parameter, which encourages the different components of the U matrix to be orthogonal. And this directly leads to a clustering viewpoint where cells can be clustered based off of which factor which factor has highest contribution. And so that's exactly what we do is we take our cell factors and our gene factor matrices and we cluster them based off of the argmax of each row. And this defines a set of cell and gene clusters where the cells in cluster 1 generally express genes in gene cluster 1. And this relationship is a one-to-one relationship, generally for NMF, but oftentimes this one-to-one relationship is too restrictive. And the reason why it's restrictive is because cells from cell type 1 and cell type 2 may share common pathways, and so we need to be able to identify these. So to overcome this challenge, we've extended non-negative matrix factorization to a non-negative matrix tri-factorization based approach, where we include this third matrix S. So this third matrix provides a few benefits. First, it allows for a variable number of genomic factors relative to the cell cluster factors, such that we can have more gene clusters that show some combinatorial relationship. Second, our S matrix readily identifies which factors are related, where the non-zero values of S indicate some sort of linear relationship between the cell factor and the gene factor. And so we can use this for clustering as well, where we, again, assign our cells to clusters based off of our U annotation. map our cell and gene clusters together based off of our relationship in the S matrix, and then determine function of our different gene clusters or gene programs based off of some sort of annotation pipeline. And we've compared SCOTCH to multiple different other embedding / clustering and tools for identifying gene factors. And so here's a brief list. ONMF is the method that I mentioned at the very beginning of the talk, where we take the original non-negative matrix factorization approach and do the bi-clustering based off of that. The other approaches largely differ in the dimensionality reduction strategy, and then have a common clustering step that uses the Leiden algorithm to cluster the cells, followed by a differential expression test. And to test SCOTCH, we looked at PBMC data set from a relatively old blend roster data set, but this data set is ideal for this test because these cells were FACS sorted prior to sampling, and so we have relatively accurate gold standard labels. And so it's a small data set consisting of 2,638 cells from these terminal cell states. And if we look at the embedding of all of the different methods relative to the gold standard, we can see that really all of the methods do a fairly good job at recapitulating the structure of the gold standard data. Next to each of the methods, we include the cell ARI, which is a metric which tells the matching between the gold standard and each of the individual clusterings. And here, SCOTCH is performing best, but relative to the other methods, the structures are relatively similar. And we would say that all of these methods are capable of clustering the PBMC cells accurately. However, SCOTCH gives us the added benefit of analyzing relationships between our cell clusters and gene clusters. And so here is that representation. So on the left, we have our cell clusters, our 6 different cell clusters, and a heat map indicating the contribution of each of the different cell types within each of those cell clusters. In the middle is our S matrix, which denotes the sharing between cell and gene cluster factors. And on the bottom are the 15 different gene factors that we have and their corresponding enrichment for either cell type annotations or biological processes. So just to highlight a few of the interesting things that are present here, we can clearly see that in cell cluster or gene cluster 14, there's a clear annotation for CD8 T cells, and this maps directly to cell cluster 6, which contains our large proportion of our CD8 T cells. We additionally see that this cluster maps to our natural killer cells, as well as a different biological process, which is shared between these two cell types, which is related to the MHC class 1 protein. And that functionality is grouped within gene program 3. And if we compare this to 2 other approaches, ONMF and Leiden, we can see that the annotation for SCOTCH is generally better in that all of our different cell types contain some sort of functional or cell type annotation. And in the case of Leiden, that's just not true where we're in the case of ONMF, that's just not true where we're missing some functional annotation. In the case of Leiden, generally our terms are overly enriched across multiple different cell clusters, and this is largely due to the sharing between different gene programs across these different cell types. So next, we looked at SCOTCH's ability to cluster single-cell ATAC-seq data, and here I'm comparing to SysTopic, which is similar in nature to NMF, where it's a latent dir site allocation-based model, and then Archer, which is largely similar to the Seurat pipeline. And again, we're looking at a different Blenrostro data set, but a PBMC data set where it was FACS sorted previously. And here, rather than having just the terminal cell types, we have all of the intermediate cell types during the differentiation trajectory. And we've clustered our data using SCOTCH and the previous methods. And again, we can see that these clusterings relatively reflect the gold standard clusters identities. Here, Archer is performing the best according to the cell ARI, but the other 2 methods are largely performing slightly worse just due to some splitting of some additional clusters. And so to show the benefit of SCOTCH for being able to identify combinatorial relationships within single-cell ATAC-seq data, I'll just highlight a few clear examples. And so the first one is this region cluster 18, which is specific to cell cluster 10. And here is our cell cluster 10, and the top markers for that region largely have accessibility only within the specific cell cluster. But if instead we look at a region cluster which is mapped to multiple different cell clusters, we can see that those cell clusters largely contain accessibility profiles. And if we go ahead and look at the annotation and functional annotation of our different region clusters, here we're focused on the accessibility of the gene promoter. We can generally characterize some functionality directly from our single cell ATAC-seq data. And if we look at our general sets of features which are shared across multiple different clusters, these accessible genes are largely associated with processes that are shared across cell types, including cell division and RNA metabolism. Whereas if we look at more specific cell types, such as our monocytes or the differentiation tree that is associated with the HSCs, we can see more specific terms related to each of these cells' biological processes, including cell differentiation, the regulation of the immune response, and some innate immune response capabilities. Finally, with the single cell ATAC-seq data, we can also look at the accessibility of motif instances across different sites and associate these with our different region clusters. And these are cell type specific as well. And so here is an example of GATA1, Tau1, which is a key marker for MEPS differentiation. And this maps directly to our cell cluster, which contains all of the MEPS. A different one that we can also highlight is our PDC set, which has enrichment for the STAT1, STAT2 complex, which is an important transcription factor in the STAT junk pathway, which is important for immune response. And so in conclusion, SCOTCH is an unsupervised learning approach for identification of cell clusters and driving genomic features. And SCOTCH is applicable to single-cell RNA-seq and single-cell ATAC-seq data and identifies high-quality clusters and genomic feature programs. And we're extending this in future work to work with multi-omic, multi-sample data set, as well as incorporating prior knowledge into the structure of SCOTCH to regularize our gene factors. And with that, I'd like to thank my lab members for contributing, especially Harmon Bosson, who is an undergraduate who worked with me pretty closely on this project. Thank you.
While we get set up for the next speaker, there's time for one more question.
Thanks for the great talk. So in your formulation, the S matrix is non-diagonal. So I was just wondering if there's like a way to tell if one factor is more important than the others. Is there a way to like order them?
Yeah, so the value of the S matrix is relatively correlated to the accessibility of, or the expression of the factor within that cluster. And so the non-zero values, higher non-zero values would relatively well indicate increased expression.